Monsters of the Mariana Trench, creatures capable of living at great depths. Chinese scientists have deciphered the genome of a ray-finned fish that lives in the deepest place of the world ocean, the Mariana Trench. To withstand pressure hundreds of times higher than atmospheric pressure and the complete absence of light. Her body underwent several serious changes at the genetic level in a fairly short time. Recent expeditions have shown that many creatures live and even thrive in this abyss. The legend of the flatfish deep-sea trenches were explored and many discovered in the early 1950s by the Soviet ship Vityaz and the Danish Galatea. The deepest place on the planet is the Challenger Deep in the Mariana Trench. Until now, the lion's share of information obtained from there belongs to expeditions more than half a century ago. In 1960, the Swiss bathyscaphe Trieste first sank to the bottom of the Challenger Deep. Right below us lay something like a flat fish, reminiscent of a flounder. It had two round eyes on top. It moved along the bottom in mucus and water and disappeared into the night. Oceanographer Jean so colorfully described his impressions. Picard, pilot of the Trieste. Scientists immediately doubted this evidence, especially since there were no cameras on board. However, journalists really liked the image of the flatfish of Trieste and they captured the imagination of the general public with it for many decades. Even some professors were misled. The legend of the flatfish resurfaced in 2012 thanks to the risky venture of director James Cameron, the third person in the world to see the bottom of the Challenger Deep from a deep-sea submersible. Cameron himself, like the participants of previous expeditions, did not notice flatfish there. The Japanese, Americans and Chinese, who set traps at the bottom of the Mariana Trench, did not find them, and Trieste's co-pilot Don Walsh later spoke less confidently about what he saw. In a 2012 article, English oceanographer from the University of Aberdeen Alan Jamieson finally debunked the myth of the Trieste flatfish. First, it is known for sure that real flatfish, such as stingrays and flounder, live in shallow waters. Secondly, it is unlikely that the submersible would land directly on a fish, according to trap statistics. With depth, the average time for the first fish to arrive at them increases and reaches 10 hours. At almost 11 kilometers, the tree stayed at the bottom for 20 minutes and had no baited traps. The main argument against is too much hydrostatic pressure. Apparently, it makes it impossible for fish to live at depths greater than 5 kilometers. But in order to exist even at this level, as it turned out, you need to significantly change the body. Pseudoleparus sets a record for a long time. Erroneous fish from the class of ray-finned fish were considered the deepest. Their species, Holcomictrinus profundissimus, was caught from 6 kilometers away. In the 1970s, the record was broken by a deep-sea Brochula abyssobrochula galathea from the same family, caught in the Puerto Rico Oceanic Trench at around 8,370 meters. However, the already mentioned Jamieson doubted this too. According to the Fish Registry, there are 17 specimens of this species of Brochula, of which only 2 were obtained at great depth. So error is possible and the deepest vertebrate creature has yet to be discovered. In the meantime, the Mariana sea slug Pseudoleparus swire is considered the record holder. In 2013, he was caught by Chinese researchers during a test descent of the bathyscaphe to a depth of 7 kilometers. In 2017, Americans raised several dozen of these fish from a depth of 8,178 meters. These are small fish up to 28 centimeters long, weighing no more than 200 grams. They have transparent skin covered with mucus, through which the internal organs are visible, and 2 small black eyes on their heads. They are completely blind and do not respond to illuminated traps. This type of Pseudoleparus is at the top of the food chain of the deep-sea part of the Mariana Trench. It has no enemies, and there is plenty of food, because there are many crustaceans at the bottom. Pseudolaparus are accompanied at depth by several species of fish from the families of eelpouts, bugs and longtails. Scientists have discovered deep-sea mutations. There is more and more evidence that the organism must be specially adapted to living at great depths, without light, in the cold. The latest methods of genome research have allowed scientists to lift the veil of secrecy here. For example, it turned out that the amount of trimethylamine oxide, a simple organic compound that helps the cell not lose shape and cope with external pressure, increases with depth in the tissues of bony fish. Such substances are called osmolytes. There is also evidence that cellular proteins lose their shape due to high pressure, and this is fatal to living beings. This means there must be a mechanism to prevent this. This is how the hypothesis about pisolets appeared, soluble substances that hold the shape of proteins or even reassemble them if they collapse. In a recent article in Nature, Chinese scientists presented the results of deciphering the genome of MARIAN pseudolaparus and compared it with the genome of the common Tanaka liparis. The two species diverged approximately 20 million years ago. The gene pool of deep-sea fish turned out to be more diverse, and their population expanded dramatically about 55,000 years ago. The genome itself is 22% larger than the Tanaka liparis genome and contains fewer mutations. One of the main features is the low metabolic rate of pseudolaparus. They literally live slowly. Their females produce fewer eggs, but they are larger. In MARIAN pseudolaparus, not the entire skeleton is ossified. For the most part it is made of cartilage. This is probably caused by a mutation in the GLA gene, which stops bone calcification prematurely. It turned out that the fish had lost several important photoreceptors. They do not distinguish colors and do not capture light. They have lost the MC1R pigmentation gene, which is why they are colorless. Coloring is now unnecessary for them. Several mutations help them improve fatty acid metabolism. Pseudolaparus was found to have 15 copies of the CAR1 gene, which regulates the synthesis of docosahexaenoic acid, one of the omega-3 fatty acids. There are mutations in the TFA and SLC9A3 genes, which are responsible for the transfer of ions and solutions from the cell. All this is clearly aimed at making the lipid membranes of cells more elastic and permeable. It is possible that some mutations in pseudolaparus increase the synthesis of trimethylamine oxide in tissues to maintain the shape of proteins. Scientists discovered another strange difference. An amino acid change occurred in the HSP90 gene, and in a very conservative area that is unchanged in humans, mice and even yeast. This gene is responsible for the synthesis of a high molecular chaperone, which, in turn, is involved in the folding of more than 200 proteins important for cellular processes. What this mutation does is not yet known. The authors of the work note that MARIAN pseudolaparus had to adapt to new living conditions in just a few million years. This is a short period of time for the evolution of vertebrates. Our new habitat, the Mariana Trench, is inhabited by numerous species of invertebrate animals, bacteria, fungi, and viruses. For example, at a depth of over 5 kilometers, sea stars of the species Phragastera benthophylla live there. Chinese scientists have deciphered the genome in their mitochondria. It is a circular DNA consisting of only a few dozen genes, but there are many copies of it in every cell of the body. In general, it appeared to be similar to the mitogenome of other sea stars, with some exceptions that are still awaiting explanation. The mitogen of amphipods, a tiny crustacean raised from a depth of almost 11 kilometers, was also studied. This species appeared 109 million years ago and evolved slowly. During its life at a depth, only a few features were discovered in its mitochondrial gene, the same as in other deep-sea species, in particular, a completely different arrangement of genes in DNA. Another discovery, at the bottom of the Challenger Deep, A colony of bacteria eating hydrocarbons was discovered.